In this video, we'll review the visual pathways in the retina, and then from the retina to the primary visual cortex. We'll touch just a little bit on the processing that goes on in the cortex, but the bulk of the lecture focuses on the processing that goes on in the retina. So we'll go through the different layers, the cells that are found there, and how light leads to a change in membrane potential and chemical communication within the retina. From there, we'll follow along our optic nerve, finding our way to the midbrain, thalamus, depending on the projection we go to, and from the thalamus to the primary visual cortex. So of course this all starts in the retina, which is where we have photoreceptors. These are modified neural cells that detect light and turn that light into a change in membrane potential. These photoreceptors then communicate with bipolar cells. The bipolar cells activate retinal ganglion cells, and the retinal ganglion cells are the projection neurons that exit the retina. So of course light enters the eye by passing through the cornea and through the pupil. It is focused by the lens, and the degree to which we stretch the lens of course depends on autonomic input to the ciliary muscles. When the ciliary muscles contract, this decreases tension on those ligaments that attach to the lens, causing it to fatten and thus focus on closer objects. So we can focus light that's closer or further away. And the whole point is to make sure that when light hits the retina, it's in focus. The retina will then detect the light in the outer nuclear layer. This contains the photoreceptors. Now the reason we call it the outer nuclear layer is because it's along the outside surface of the retina. So the outer nuclear layer is going to be here. We have rods and we have cones. These are our photoreceptors. Rods will pick up dim light of any color. Cones will pick up very specific wavelengths of light, but they require a higher brightness to do so because they're filtering out some of the wavelengths. In the center of our visual field, we find a whole lot more cones, and the rods are going to be more concentrated on the outside. But I have my outer nuclear layer around the back. Then I have the inner nuclear layer. This contains interneurons, one of which being the horizontal cell. This is going to provide a little bit of feedback onto photoreceptors. We'll touch on that later. We also have the bipolar cell. Now this is the interneuron that will communicate with our retinal ganglion cell in the retinal ganglion cell layer, and notice I've made my retina incredibly thick here. Right, it should only be a couple hundred microns. And then we have amacrine cells. And amacrine cells are fairly complicated, so we won't really talk about them. They release every neurotransmitter under the sun. There's probably at least a couple dozen types of amacrine cells, but these are local interneurons within the inner nuclear layer. They'll talk to bipolar cells, other amacrine cells, and ganglion cells to affect their function. The last layer would be the ganglion cell layer, and that's actually closest to the light. So light would enter, get the iris up here. We'll focus. Let's get some ciliary muscles, some suspensory ligaments. That light gets focused and absorbed back here. Retinal ganglion cell layer contains the retinal ganglion cells. These are the projection neurons that have to go back through the retina and exit. As such, wherever our retinal ganglion cells are exiting, we can't put photoreceptors. The site where we exit is called the optic disc. we can see this in our vision because it creates a blind spot. Of course, we don't actually see the blind spot unless we go looking for it, because the cortex paints over the blind spot. But we do all have a little blind spot, just a little off of the center of our vision, where we have no photoreceptors, so we can't pick up any light. The reason we have that is that all the axons of the retinal ganglion cells had to exit somewhere. Just keep in mind, that outer nuclear layer and the inner nuclear layer are between the retinal ganglion cell layer and the brain. So we pierce here at the optic disc to create our optic nerve, second cranial nerve. Alright, let's consider what's going on in all of these cells to allow us to have light translated into activity of the optic nerve. Well, it of course starts with photoreceptors. Photoreceptors contain photopigments. This is a combination of retinal and opsin. Retinal is the pigmented molecule that absorbs light. Opsin is a G protein coupled receptor. Cones are color sensitive because they express 3 different types of opsins. And depending on the type of opsin, that affects which wavelength of light retinal absorbs. So it changes the color of retinal. L opsins pick up kind of red, yellow type light. M opsins pick up green and yellow. And then S opsins pick up more of the purples and blues. That's the cartoon representation. Of course, in real life, they pick up a spectrum of colors, as you can see over here. So look in panel A. This shows you the spectral frequencies for the S, M, and L cones. You'll notice S is quite distinctive from M and L, but M and L have a lot of overlap. Sure, L is right shifted and will pick up more of the red, but they overlap considerably on the greens. Despite this, we simplify them, as you can see in panel B below there. S cones we call blue, M cones we call green, and L cones we call red. Now rods don't have any sort of special opsins. They express a color-insensitive opsin called rhodopsin. This causes retinal to absorb a variety of wavelengths, making rods color-insensitive. Now, regardless of which opsin you express, the same thing is going to happen under the hood. When light is absorbed by retinal, this activates opsin. Opsin is a G protein coupled receptor, and the G protein that's attached to it is transducin. So we'll just get us a rod up here. So we've got some sort of pigmented molecule over here, something with color. That's my retinal. Here's the opsin. And I have GT. Now I know we didn't talk about this one earlier, but I did mention that there would be additional G proteins. Transducin is kind of like GI, except it goes about decreasing cyclic nucleotides in a different way. This stimulates an enzyme called phosphodiesterase. Phosphodiesterase is an enzyme that breaks apart phosphodiester bonds. And what it does is turn cyclic GMP into just regular old GMP. So when light is absorbed, transducin is activated. That stimulates phosphodiesterase, and we start breaking down cyclic GMP. So light causes us to decrease cyclic GMP levels. Well, let's not forget this is a cyclic nucleotide. Normally, cyclic GMP stimulates those cyclic nucleotide gated cation channels. So if we have a lot of cyclic GMP, that allows a whole lot of positive charge to flow in and keeps my membrane potential fairly depolarized. of course, when light hits, we're breaking down cyclic GMP. As such, we hyperpolarize the membrane potential. So my rod and my cone is actually less active when light is around. When I hyperpolarize, this of course decreases glutamate release. Now normally, I'm fairly depolarized and I'm spitting out a whole lot of glutamate. So photoreceptors have those ribbon synapses, just like we find in the inner ear. So those hair cells can always spit out glutamate and keep us informed about sounds and head position. So too can photoreceptors always tell us what we see. So they have these ribbon synapses that increase the readily releasable pool and allow them to keep up with the fairly high rate of glutamate release. About 20 to 80 or so vesicles being fused per second. So we would quickly run out without a good old ribbon synapse, increasing the size of our readily releasable pool. So normally we're just constantly spitting out glutamate until we hit with light. So let's just put dark over here. We're spitting out a lot of glutamate. With light, we hyperpolarize, don't spit out as much glutamate, and that's what's going to be detected by the rest of the retinal circuitry, a decrease in glutamate. Now this is not all or none. These photoreceptors don't fire action potentials, instead there's just a graded release of glutamate, depending on how depolarized we are. If we're more depolarized, of course we open up additional voltage-gated calcium channels, more calcium, more glutamate release. Light breaks down cyclic GMP and hyperpolarizes us, leading to a decrease in calcium influx and thus a decrease in glutamate release. Now the distribution of photoreceptors is not even throughout the retina. In the center of our visual field, this corresponds to the portion of the retina called the fovea. So whatever you're looking at, that light is hitting your fovea. And whatever's in the dead center is in the foveola. So that's the center of the fovea. In the fovea, we have a very dense collection of photoreceptors. And that gives us the best visual acuity, because each photoreceptor is smaller. They have a smaller receptive field, and so it's easier for us to distinguish fine details. This is kind of like the two-point discrimination difference between, let's say, our lips, which have a high density of innervation, and our back, which have a low density. So the high density of very small photoreceptors in our fovea allows us to distinguish fine details. In the periphery, we don't have a high concentration of cones. Instead, it's replaced by rods. Now, there's a difference between cones and rods, not only their location, but also their coupling to retinal ganglion cells. Cones, for the most part, are coupled 1-to-1. So we have a cone, some bipolar cell, and then a retinal ganglion cell that'll project away. Rods, on the other hand, will converge, and imagine that I've drawn, 15 or so of those. So there will be multiple bipolar cells, and all of these will converge onto a single retinal ganglion cell. So for cones, it's 1-to-1. For rods, it's 15 to 30 or so to one retinal ganglion cell. So the retinal ganglion cell that gets input from rods is sampling a much larger region of the retina. This has two effects. It decreases visual acuity. so we don't see as fine of details. But it makes us far more sensitive to dim light because we're sampling a larger area of the retina. Now in that foveola, we only have actually the outer nuclear layer. We get rid of those bipolar cells and those retinal ganglion cells. We scoot them out of the way. So that's what this illustration is showing us here. So on top you can see the pink and green rods and cones there. And in the foveola, the layers separate, like parting the waters, and light can directly stimulate the outer nuclear layer without having to go through all those other cells, potentially getting a bit scattered. This also increases our visual acuity. So there's two things going on. Get the other layers out of the way and pack in as many photoreceptors as you have. As you can. In order to do that, the photoreceptors need to be a little bit smaller. So these are what we call midget cones. These are much smaller than typical cones you find outside of the foveola. So in the rest of the fovea, the cones are a little bit larger, meaning that they sample a slightly larger area of the retina, decreasing visual acuity. This illustration summarizes the location of rods and cones in the retina. If you'll look on top or bottom, it's the same thing. And what they're showing us is that rods are located more so in the periphery. So here we're looking at the distance from the fovea. That's basically what our x-axis is. Call 0 the fovea there. So of course that contains an awful lot of cones. Rods we see a lot more commonly in the periphery compared to in the fovea. And then of course we have to keep into account that blind spot, the optic disc. So that's showing up in this illustration as well. You can't put any photoreceptors where you're running all of your retinal ganglion cell axons. But because of this distribution, where we find rods in the periphery, this makes our peripheral vision a little less sensitive to color. So we're fairly colorblind, in fact, in our periphery. We don't get that impression because, again, our cortex does a good job of painting over the kind of dim colors that are actually created in our retina. So it gets rid of the blind spot, and it makes us think that our peripheral vision isn't as colorblind as it is. Moving on, we get our bipolar cells. So we got rods and cones. Rods and cones talk to bipolar cells. So just like there are two types of photoreceptors, there are two types of bipolar cells, on and off. Off bipolar cells, as the name suggests, are turned off by light. The reason for this is that they're turned on by glutamate. On bipolar cells, on the other hand, are turned on by light. And the reason for that is that they're turned off by glutamate. Let's not forget, light causes a decrease in glutamate release from photoreceptors. So, the reason for this difference has to do with the type of glutamate receptor that they have. Off bipolar cells have good old ionotropic glutamate receptors, AMPA kainate receptors. So when glutamate binds, positive charge moves into the cell. Bipolar cells depolarize. Of course, when light hits the photoreceptor, it doesn't spit out as much glutamate. So we don't have as much of a cation conductance. Thus, we're not as depolarized. You only find off bipolar cells attached to cones. Rods don't have them. There is no need. The reason we use them with cones is because we have to take into account the absence of certain wavelengths of light when we process colors. More on this later. On bipolar cells are turned off by glutamate because they have metabotropic glutamate receptors that are GI coupled. GI, of course, decreases cyclic AMP, not as much cyclic nucleotides, we decrease cyclic nucleotide-gated cation channel conductance. Thus, glutamate normally hyperpolarizes us, so the loss of glutamate depolarizes us. Bipolar cells then communicate with amacrine cells, which we won't talk about, and retinal ganglion cells. But before we can move out of the internuclear layer, we need to discuss horizontal cells. Horizontal cells aid in edge detection, and they do so by providing negative feedback to photoreceptors. So the basic circuit here is photoreceptor, horizontal cell, bipolar cell, and we'll eventually go to a retinal ganglion cell. But we're not there yet. What horizontal cells do is provide lateral inhibition. They do this by releasing GABA. So if I hit light here, but not here, so let's consider a couple of neighbors. That light causes hyperpolarization of this photoreceptor. Now you might think that that would have no effect on the neighbors, but that's not the case. We have horizontal cells. So we're not spitting out as much glutamate. As such, these horizontal cells also hyperpolarize. So they don't spit out as much GABA. If we're not inhibiting these photoreceptors as much, they actually exhibit depolarization. So notice, right here at the edge where I have light and dark, at that boundary, I create a bigger contrast in photoreceptor activity. That makes the light appear a little bit lighter and the dark appear a little bit darker. I could draw this out a little further, but this tells the story. Now sure, they've got their horizontal cells getting input from bipolar cells, but the story is the same. On top, what they're showing is just a change in the intensity of light. So that's that top line there. It's going from dark to light. So at the edge, what's going on there? Well, there's a bigger change in activity if you're in the light there than if you're in the dark. Okay, so those photoreceptors should be getting hyperpolarized. It's going to go through the retinal circuitry, and on the bottom it's showing you the output. So what's going on there? Okay, let's walk through this a little bit. So here we just have to put some fake numbers down so we can walk through the math. So where I have my light, let's say that this hyperpolarizes me by, I don't know, 10 millivolts or so. And we'll just keep the dark as, no change. Okay. So I'm not spitting out as much glutamate over here as I am over here. Let's not forget our horizontal cells. Okay, so there's a little lateral inhibition here. Now that glutamate that's getting spat out over here in the dark is keeping these horizontal cells fairly active. So let's think about how much stimulation we're giving these. The numbers don't matter. Hell, let's call it 2, because I'm getting input from, let's say, 2 cones. This one's getting input from 2 cones. This one's getting strong input from 2 cones. This one's only getting strong input from 1 cone. Right? This one's providing, but this one isn't. This one's not getting input from any of these. Okay. So now, how does that feedback affect my photoreceptors? Well, I'm going going to inhibit over here, but there's a bit of a difference right here at this edge. Notice I'm not going to quite provide that much inhibition over here. So let's call my feedback here nothing. Okay, so we'll subtract nothing for these. But I'm actually still getting a little bit of GABA feedback over here. So I got to subtract, we'll say 1 here. Right, 1 and 0. This one, it's getting, let's say 2 units of GABA plus 1, so now I got to subtract 3, and here I'll subtract 4, and 4, and 4. So notice I'm creating some contrast here. Right here at the edge. Alright, how does that affect then my communication with my retinal ganglion cell? Well, we got to go through a bipolar cell. Let's just assume some one-to-one coupling here. Alright, and we'll make all these on. So how on am I depends on how little glutamate I'm spitting out. So I'm spitting out -4, so I'm just going to change the sign. I'm going from inhibition now to excitation. So if I'm spitting out -4, we'll call that now +4. +4. +4, only +3. +11, +10, +10, you get the point there. Okay, so the real difference that I have, if I just look at the 4 to 10, I have about a 6 unit difference in activity, except at the edge. Here I have a bigger difference. I have an 8 unit difference in activity. So the contrast in output here and here is much greater than the contrast in output for the rest of the light and dark zones. You'll notice at the edge we have much greater contrast. So horizontal cells help edges pop. We'll see an example of this in just a little bit. The other thing that horizontal cells do is create center-surround organization in our retinal ganglion cells. So the last little bit here, that's what this is going to feed onto. Rather than draw that out, let's go through our center-surround. Why do we have center-surround organization? Well, let's go back to our little circuit here. Cone, bipolar cell, retinal ganglion cell. So now we need to think about the receptive field for this retinal ganglion cell. Where does it get input from? This cone, but actually it also is going to get some input from the neighbors too. Let's not forget, because we have horizontal cells. Alright, let's say we shine light in the center, just like we did before. We hyperpolarize this cone, it dumps out less glutamate, that turns on our bipolar cell, and thus we turn on the retinal ganglion cell. So hey, I'm on when light's in the center. Now of course, if this were an off bipolar cell, I'd get the opposite. If I spit out less glutamate, off bipolar cells aren't as active, retinal ganglion cells not as active. Okay. But let's just stick with on for the ease of comparing center and surround. So that's light in the center. What if I put light over here in the surround onto a neighbor? Well, that one's going to hyperpolarize. Well, that means that this horizontal cell isn't going to be as active which means that actually this photoreceptor depolarizes a little bit, dumping out a little more glutamate. That, of course, turns off the on bipolar cell and turns off the retinal ganglion cell. And that explains what we see over here in the center surround in organization. So if you look at B, they shine light on the receptive field. Let me rotate this 90 degrees. What that would look like is that cone, so that'd be the center, and then in the surround we'd have all the neighboring cones. So I'll just put some dots for them. So all the nearby cones and the one in the center. Okay, so if we shine light here, we see an increase in retinal ganglion cell activity while the light is on, if it's in the center. If we shine light in the surround, we see a decrease in retinal ganglion cell activity. And that's created, again, by those horizontal cells. What's also created by horizontal cells is this illusion, the Hermann grid. So if you have a look at this grid, what you'll see is that wherever you're looking, you can see that it's uniform white between all those black squares. But surrounding that, at the intersection, it looks a little bit darker. What's going on there? Differences in center surround. Let's think about our receptive fields, right? So when you're looking at the intersection, remember in your foveola, very small photoreceptors, very small receptive field sizes. But in the periphery, the receptive field gets a little bit larger. So when we are in, let's say, a straight edge of this grid, let's think about how much light is entering the center versus the surround. So I put up two receptive fields there, blue for the center, red for the surround. So we got our squares. So if you're looking over here, this is going to appear a little bit darker. And the reason for that is we're doing that comparison between center and surround. Notice we're getting light here, 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 and here in our surround. Now what about this area right next to it? Well, what that retinal ganglion cell sees is this. I only have 2 bits of light in my surround, so I'm not inhibited as much. The activity of this retinal ganglion cell is higher than this one. As such, the brightness that we detect here is lower at the intersection, and that's simply because of center surround. After we've made it through our photoreceptors, bipolar cells, and of course the horizontal cell filter there, retinal ganglion cells then project out of the retina and communicate with the brainstem and thalamus. From thalamus, we go to the cortex. So the retinal ganglion cells make the optic nerves. The optic nerves come together to form the optic chiasm, and this is where axons from one eye can cross over. Because the way that we organize our visual system is contralateral to our visual fields, not our eyes. So whatever's in the left part of the world goes to my right visual cortex. It doesn't matter which eye it hits. Whatever's on the left hits the right side of my left retina and my right retina. Those two right sides head on back to the right side of my cortex, as this illustration shows. So the left eye there is colored blue, the right eye is colored red, and you see they're crossing over at the optic chiasm. After crossing over, we now call it the optic tracts, even though nothing has really changed. The optic tracts have a medial root that innervates the midbrain, serving the superior colliculus to help create some automatic eye movements if needed. The pretectal nucleus to assist in the pupillary light reflex, and then the reticular formation to provide input about light. The lateral root is what we'll concern ourselves with for the rest of this lecture. That lateral root is going to go on to the lateral geniculate nucleus of the thalamus. The lateral geniculate nucleus contains distinct layers of cells that respond to different types of retinal ganglion cells. The first two magnocellular layers there receive input from M cells, M for magnocellular. M cells are retinal ganglion cells that get input from rods. The M cells are thus not very responsive to different colors, but they are very much responsive to dim light. And because M cells rapidly desensitize, they're also sensitive to movement. So M cells pick up dim light and little bits of movement in the periphery. Not fine detail, but enough to see that if something's moving toward us. For finer details, we want input from the P and K cells. P cells project to the parvocellular layers. Parvocellular, of course, meaning small cell. P cells are retinal ganglion cells that get input from cones that express M and L opsins. So they're sensitive to red and green light. These also have fairly small receptive fields and display your typical center-surround organization. K cells project to the coniocellular layers, and those are not labeled in this illustration. Instead, they are the light bands you find between the magnocellular and parvocellular layers. Coniocellular just means dust cell, so these are even smaller than parvocellular neurons. They're still color responsive, but they are responsive to blue light rather than red and green. Thus they get input from photoreceptors that express S opsins. K cells have larger receptive fields, and they only have center, no surround. So they don't have that surround inhibition. Because of this, our ability to pick up blue light is a little worse, especially at night. You might notice that blue light appears a little fuzzier. We don't have that filtering at the edges provided by horizontal cells. And the slightly larger receptive field also decreases visual acuity. Now, unfortunately, we have other naming that you might run across for retinal ganglion cells. There are midget cells, which are small, and parasol cells, which are much larger. These are named after their dendritic arbors. Parasol cells have a large dendritic arbor that I guess kind of looks like an umbrella over them. Now you might think, midget, M, that's the M cell. No. Those are the P cells. Parasol cells, hey, P. No, those are the M cells. It's unfortunate, but now you know about it. So we project to different parts of the lateral geniculate nucleus depending on what kind of information we work with. Rods go to the magnocellular layer. If you get input from cones, you're going to go to either the parvocellular layers, if you respond to red-green, or the coniocellular layers, if you respond to blue. From the lateral geniculate nucleus, we then project along the optic radiations to the primary visual cortex. The primary visual cortex is, of course, in the occipital lobes. Within the primary visual cortex, each column of cells, so that is all the different layers of the cortex in any given location. So if this is my primary visual cortex, we got different layers 1 contains no cells. 2 and 3 of the cortex has little projection neurons. Layer 4 is the big one for us here, because this gets thalamic input. 5, deep projection. 6, feedback to the thalamus. But in any given column of cells, so all the different neurons that we find, let me put a few little neurons in here. I'm not going to connect them to anything, but just so we have something. All of these cells respond to input from one eye, and they alternate. Left, right, left, right. These are what we call ocular dominance columns. So although both eyes will project to the right or left primary visual cortex, within the cortex we keep our eyes separated in the primary visual cortex. And this likely aids in detecting disparity between the two eyes. And those little differences in how things look in our eyes helps us determine depth of objects. You'll notice if you close one eye and the other, what you're seeing looks a little bit different. And so we learn to translate that into depths. Now, even though we're separating our eyes, we're still maintaining an organization of the retina. So we still have a retinotopic map. You take that retina, you can project it pretty well onto the primary visual cortex. That's what they're showing you colored here. So the middle of that is the calcarine sulcus. On both sides we have a retinotopic map. And they numbered it on top. Fovea in the center, darkest color, you'll notice occupying the largest area of the cortex, even though it's a small area of the retina. That's because of the density of innervation. More photoreceptors and more retinal ganglion cells corresponding to the fovea corresponds to a greater number of cortical neurons that correspond to the fovea. Within the primary visual cortex, we deal with very granular and bitsy information. We don't see beyond very fine details. You may have heard you can't see the forest for the trees. We're not even looking at trees at this point. We're looking at little bits of bark in the primary visual cortex. So we're sensitive to lines that are this angle, or this angle, or this angle. The line might have to stop to turn on a neuron, or the line might have to not stop. So we're looking for very fine details. Do I have any edges? That is, do lines stop? And what are the angles of all the lines? We're going to put together higher order information by walking through the cortex, like that schematic shows us on top. So we'll go from the primary visual cortex, V1, to secondary, V2. We'll go to other association cortices, V3, V4, V5. You get the point. We'll even go outside of visual cortices to ultimately determine what we're looking at, such as faces or putting a name to an object. But we start very small. We pick out all the angles of lines. So we have lines like this. We've detected these. Now we want to think about how are they related to each other. So in secondary cortices, now we're going to say, okay, well, they were oriented as such. All right, now we can pick out a shape. We don't pick out shapes, though, in the primary visual cortex. We just pick out lines. Then we build shapes later. And we start to also take into account things like color. I'll tell you that this is red. And there's some curvy junk in there. Well, okay, we're going to move on to other higher level association cortices, and we'll finally figure out, okay, we got an octagon that's colored red and it says STOP in it. That's a stop sign. But the primary visual cortex doesn't know this. All it sees is lines, just fine details, and it tells its neighbor in this kind of assembly line of perception the angles that it sees, and whether they ended or not, and where they ended. Somebody else can figure out what the heck I'm looking at. We also need to figure out color. And this is partly done in the retina, but not that much. We do have some color opponent cells. Color opponent meaning that two colors actually oppose one another. Okay, so we got three cones, right? S, M, and L. We'll say blue, green, and red. How can I pick out blue light? Well, I could just directly stimulate. So let's say I have an on bipolar cell here, and I go to a retinal ganglion cell. Cool. But how can I distinguish blue from white? White light contains blue in it. It'll certainly have all the wavelengths to turn this on. So without any color opponency, I can't distinguish colors from white light. I need to have some kind of input here. So there must be convergence. That's why for the most part this happens in the lateral geniculate nucleus, not the retina. Because coupling is typically 1-to-1. We do have some convergence in the retina, but it's rare. You should think of color opponency as being more a function of the thalamus and a bit in the primary visual cortex. But we'll just assume that there's convergence for the sake of this cartoon. And in some cases there are. Well, okay, if I'm going to be blue light and not white light, that means I have to have blue, but not green and not red. If I only have blue, I'm blue light. So that means I need to have this kind of circuit. If I have blue light, I will stimulate the S cone, but not the M and L. That means I'm still spitting out glutamate over here, but let's just say that I'm not on this one. That of course turns on the on bipolar cell. The presence of glutamate turns on the off bipolar cells. So I get very strong input. And I have maximal activity of this retinal ganglion cell, or more likely this parvocellular, well no, coniocellular neuron in the LGN, or some neuron in the primary visual cortex. On the other hand, if I shine white light on here and stimulate all of these, well, okay, no glutamate from any of them. I'm turning off my off bipolar, and now I get much weaker activation of the retinal ganglion cell. So I made a little cartoon that kind of shows the wiring diagram. If it's a blunt arrow, that means there's an off bipolar cell. If there's a pointy arrow, it's an on. So blue-yellow, that would be this. This is a blue on, yellow off. Let's say I shine just yellow light. Well, that gets M and L. Doesn't stimulate this. This is off. So yellow light gives me the least activity. Another common type of color opponency is red-green. So in this case, we don't actually care about this. So this would be for like the parvocellular layer here. So if I want to be green on, that's pretty simple. Here's your wiring diagram. If I have green light, this cone is hyperpolarized, no glutamate coming out. This one is still depolarized. My on is active, my off is also active over here, and so I have maximal activity of the retinal ganglion cell. Thus it's green on. On the other hand, if I shine red light, that's going to hyperpolarize this cone, not this one. So I turn off this on bipolar cell, and I turn off this off bipolar cell, giving me red off. And so you can see why we only use off bipolar cells for color opponency. It's the only time you need it. For rods, we're just picking up light. Now, we do a little bit of processing in the retina, but it's fairly gross. Broad distinctions between colors. Color opponency is more a function of the lateral geniculate nucleus, and the primary visual cortex takes it one step further. So in the LGN we have our red on, green off, blue on, yellow off, and those neurons then converge on primary visual neurons so they can determine, well, about how green and not red are you, right? but they're not picking out the hue until we get to the secondary visual cortex, and that's where we distinguish all those millions of colors that we can pick up. Hopefully that made a little bit of sense. If anything needs further clarification, please enter that in the questions box so I know what to talk about in class. See you later.